So thank you very much for the introduction, and I'd like to talk to you about some of my work regarding epigenetic programming of early life stress. And so it's a bit of a background to a lot of the work that I'm doing, and some of the work I do does involve next generation sequencing as well. And I'd also like to, if I've got time at the end, to discuss some of my current ideas about how you could use next generation sequencing in my particular area of work. So a bit of the overview then, I'll go through a bit of the background to my major area, which is really the hypothalamic pituitary adrenal axis and stress. And I'd go through an intergenerational rat study I'm working on. And a major part of my talk, I'd like to really discuss about the idea about how you're able to use animal models and human studies at the same time as a part of a translational approach. So stress obviously means different things to different people, but the whole idea is that stress is regulated through a particular endocrine mechanism that we have. So we have the hypothalamus, which is connected to the pituitary. Now, hypothalamus gets signals from various parts of the brain in response to various stresses that we may perceive, and that stimulates the hypothalamus to stimulate the pituitary to release vasopressin and CRH, and this acts at the adrenal gland, and the adrenal gland will release this stress steroid here called cortisol or corticosterone in animal models. Now, corticosterone or cortisol is the important stress steroid, and it readies our body for like the fight or flight response, for example. Critically, it also has a negative feedback inhibition. So a bit like a thermostat, when our stress systems get activated and our perceived stress is no longer there, it will feedback inhibit and shut itself off. Now, one of the major things of stress, of course, is some stress is good. People often think stress is bad, but stress is good. So depending if you're quite scared of spiders like I am, this is quite a harmless house spider we have here. Stress enables me to leave the bathroom within about half a second without touching the floor. Obviously, spiders are not that sort of stress for other people. So these are sort of the very sort of acute stresses, allows us to remove ourselves from a situation or to handle the situation very sort of quickly. These stresses are nearly always bad, and this is sort of the stress from sort of chronic stress. So obviously paper isn't gonna kill us. We're not going to drown in the sea of paper, but psychologically we can perceive that as a major sort of stressor, and this is the sort of stress that can become very bad if we're not controlling this. What happens with a lot of stress is that under chronic stress, sometimes our feedback inhibition is not working. So we're constantly producing more of the vasopressin CRH and then the ACTH and then the cortisol, and we're not able to shut ourselves off. And a lot of people with major depression have a defect in this negative feedback inhibition. So how is this stress system activated and actually controlled? So the whole theory really is that our stress system is really built during early life, that we adapt to how the stress system we perceive we will need later on in life. So if we're expecting a high level of stress later on in life, we may well develop a stress system to enable us to handle that later on. An old quote by a very famous philosopher was "childhood decides," and I think with epigenetics and many diseases, childhood does decide an awful lot of this. So we're looking at epigenetics. So epigenetics really are the markers that can be added onto the genome to allow a genome to be differently read and to differently function in regard to the expression of various genes. So denomethylation can inhibit gene expression. So if you have high levels of denomethylation, that can recruit methylation binding domain proteins and chromatin remodeling and can shut a gene off. If that methylation is removed, transcription factors can bind and that gene can become active again. And a whole host of systems involved in methylating and demethylating the genome. So the whole hypothesis, firstly, was that early life stress could epigenetically program genes and expression of genes involved in regulating stress. So the whole idea is that early in adaptation, we have key time points where we are highly sensitive to the environment. And what happens is that we adapt through epigenetic changes in key parts of the genome, and that programs our endocrine circuits to give us an endocrine phenotype which is perfectly adapted to how we expect our later environment to be. Whether it's well adapted means that it's a good fit for our later environment. Of course, it could also be maladapted. We could have high levels of stress in early childhood, and you may never see those levels of stress again later in adulthood. So what happens is we've got a stress system which is overactive. So even under relatively less stressful conditions, our stress system is still overactive, and we're producing that level of stress, anxiety, and that of course leads to mood disorders such as depression. So translational approach really is using animal models to investigate and to highlight hypotheses that we test in humans, and also using human studies that we can test mechanisms hypotheses in animals. So they both have key important aspects which should be translationable between them. So one of the first things I was looking at was separating mice from the mothers as a stressor. So if I separate a mice pups from the mother for about 3 hours for the first 10 days of their life, so they still receive all of the nutrition that they need, but they simply don't have the mother with them for around 2 to 3 hours each day. What happens is when those mice grow up, even 1-year-old mice produce more corticosterone, as I said, the cortisol. This is the stress steroid. More of this than the control mice, even after 1 year. So they are permanently producing high levels of cortisol, and also the hypothalamus, vasopressin, and the pituitary, the ACTH gene, are all upregulated. So the HPA axis has been basically permanently changed by the early life stress, and that programs increased stress response. And these animals also show increased depression and anxiety at 1 year as well. So we looked at methylation, and methylation changes were found in the vasopressin gene in the PVN only at those neurons important in regulating stress. So other neurons also express that gene, but they're involved in a different function, mainly to regulate body fluids, and it's not changed there. So what we have is we have very cell-specific DNA methylation changes in the hypothalamus. Now, what these DNA methylation changes we see are, we have with stress response, those animals exposed to those 2-3 hours without their mother, we see less methylation than in the control. That less methylation gives more vasopressin gives higher HPX activity and higher stress responsivity. The way that's recruited is through an MECP2 binding, and MECP2 is specifically targeted and specifically activated in those neurons I showed you in the PVN. And also what I was able to do is to show how this MECP2 was recruited through a whole host of other boring chip experiments. There were hundreds of chip experiments here, in which polycomb, first of all, rests at this region in the vasopressin enhancer, and during a postnatal period, it recruits MECP2, and MECP2 is differently altered by the stress, and then we get a programming of this methylation program here. So how is this stress given to the pups? How do the pups actually receive this stress simply by being separated from the mother? So this led me to the other hypothesis, really, that stress is transmitted through alterations in the maternal care. So you need high levels of maternal care there in order to regulate the stress response. And if we don't have the mother there, such as through the separation, then we get the alteration in the stress response. So developed a rat model of transgenerational changes in maternal care. So what we have is we have a rat, we have the mother rat, and we give her a social stressor. So we have a male rat in the next cage. The female mother then prenatally and also postnatally, we've done a number of experiments, but for example, postnatally, the mother then is unable to look after her pups as well. So she feels the stressor, she doesn't lick or groom them as much, and those pups then also take on that stressor. So what we did is we did that. We measured measures of maternal care and depression in the mothers and in the infants, and we looked at endocrine and immune changes and all those hypothesized mechanisms which we think could be important. So by doing this, what we saw is that the social stressor in the mother reduced her licking and grooming of the pups, it reduced her aggression, it increased her restlessness, it had no effect on the milk giving, But what happened was that those pups sensed that reduced levels of maternal care, and they then expressed stress-related behaviors. They showed stress in their neuroendocrine systems and in gene regulation and epigenetic changes as well. So by looking at that, then we wondered then if this could translate to humans. So also particularly important, I showed you, was the licking and grooming as well. And that's a big thing that was definitely dampened by that social stress. So going into humans then, we looked at mothers, and if they're also having skin-to-skin contact, just like the rats would have during early postnatal life as well. And looking at the real study, so this is a study of 1,200 children in Liverpool, and their interactions with the parents, the maternal care. And what we see here is that those mothers with low maternal care, so with increasing levels of maternal maternal depression here, so those mothers with high levels of maternal depression, they show infants with high levels of stress. So it's well known that the levels of maternal depression, which is what you're sort of inducing here with the social stressor, those mothers with high levels of stress and maternal depression, those infants basically inherit those levels of stress, as we see here, just like in the animal model. Now, interestingly, those mothers that stroked more, they actually dampened that effect. So the actual stroking has been seen, just like in the animal model, is highly important for regulating early life stress. And this can be through C-tactile neurons, for example, which are within our skin. And that's something I'm looking at the moment. And that the idea that these could release oxytocin, important in bonding. And so the whole idea was that early life stroking actually then also changed the methylation, just like in the animal model I was looking at. And what we can see is that by stroking, you actually have a reduction in the glucocorticoid receptor. And that's the receptor in the brain that binds cortisol and is important in the negative feedback inhibition. Now, another thing we then looked at in the human studies, so although we were able to show in the human studies findings we found in the animal, what we found in the human study was that we also have this adaptive response between this mismatch and adaptive response between prenatal and postnatal. So if you know the Barker hypothesis, the idea is that we adapt ourselves to the later environment, as I mentioned before. So what we looked at is the prenatal and the postnatal adaptation. So the whole idea was that if you've got high levels of prenatal stress before you're born, that may adapt you to levels of postnatal stress. So by looking at that, what you found is that those infants with the highest levels of postnatal stress that didn't have the prenatal stress, they showed the high levels of anxiety and stress behavior. Whereas those infants that had been exposed to prenatal stress didn't show high levels of stress and anxiety following postnatal stress. So that prenatal stress allows you to adapt to a more stressful postnatal environment, so you're not showing those more extreme responses. And so that's what a study I'm working at the moment, which just getting data from due to COVID lockdown, where you got the prenatal and the postnatal. So the whole idea is that those animals without the prenatal stressor, but just the postnatal stressor, will show the highest levels of depression. And those with the prenatal stressor and the postnatal stressor will show lower levels as they've already adapted to the prenatal stressor to expect the postnatal. And so the whole idea was that the stress could then be carried through through generations of the animals. So the whole idea then is that if you have mothers with levels of stress, I'm just checking my time, then we would see if these could be inherited over more than one generation, even 2 to 3 generations. Yes.
Don't worry about the time. We are all at your disposal. It's your time.
Okay, thank you. So I just realised that the clock I hadn't on my screen has gone off with the timing.
I'm enjoying that.
Okay, thank you. So the whole idea was that I've seen that stress from parents, so the mother, and in the real study, the mother in the real study, humans and animals, that stress can lead to increases of stress and depression in just the F1 animal. But what about the F2? What about the F3? Can you see generational changes just through having a grandparent who's had that level of stress? And that would make sense, really, if we're adapting as a species, if we're adapting. So if you're looking at epigenetic, behavioral, endocrine and immune changes, milk, microbiome, any sort of aspect of a mechanism we could think of that could be important in this. So what we've done is we have the mother during after she's given birth. We give a chronic social stress to the mother. So the infants aren't affected by the stress, just the mother has, again, a male in a separate cage, but a cage nearby, and it upsets her. Then what you do is you grow that pup to be a mother. We check her pups to be and then also as mothers as well. So we've got 3 generations going on here. And what we find, amazingly, is within the FO, we see the pup grooming again, which I said was important in regulating stress. So we see reductions in pup grooming in that mother exposed to the social stressor, but just at day 9. So first of all, she handles it, then she has a problem with the social stressor, but amazingly, she adapts. And at the end of that of the postnatal period, she actually has perfectly normal grooming again. So it's amazing how the animals adapt. Now, the F1 animal, who was exposed to less grooming at day 9 from its mother, they show reduced grooming at day 2, but they regain the levels of grooming as well, so they also adapt. But at the F2, you'd first of all think that this stressor in FO has been lost, it's been forgotten, but it hasn't been. It's actually exasperated, so it actually is increased. So in the F2 animals, which are exposed to low levels of grooming at day 2, they show lower levels of licking and grooming to their pups at day 2, 9, 16. So the whole level of maternal care has changed in these animals and going forward as well. So it's quite amazing how within 1 generation, you've seen a permanent shift. So what we have is within the FO to the F2, we have massive changes in maternal care. So it's not just pup grooming as well, it's also retrieving the pups as well, and a number of other aspects. We have increased levels of maternal restlessness, which is usually associated with maternal depression and stress. So that's massively increased in F2 animals, far more than it was in the FO. And then we also see some changes in milk intake at the beginning, but we see no changes in the F2. So it's not a nutritional effect. They've basically shifted their maternal behavior completely. Interestingly, in the F1, we see increased levels of cortisol or corticosteroid. In the F2, these are decreased, so you've got slightly reduced levels. We also see changes in gene regulation through particularly genes involved in HPA axis as well. And we also see changes in some immune factors as well, particularly ICAM1 and a number of other factors. Particularly ICAM1, we see more of a shift in the F2, which wasn't there. So some things have very much changed in the F2 generation. So looking at the glucocorticoid receptor, so glucocorticoid receptor is well known to, as I said, which I've shown you in the previous animal model, to be important in regulating the HPA axis. So this is important in that negative feedback. When you've got too much cortisol, the glucocorticoid receptor allows that to turn off. So what we see changes in the methylation and the expression of the glucocorticoid receptor within the brains of these animals at the F1, but at the F2, they're actually normalized again, which is really strange. So we're having another mechanism coming in at the F2, which is somehow changing the whole stress and behavior in these animals, but it's not running through the HPA axis anymore. It's something else. Now, I would have loved to have shown you the microbiome, which sadly is just sat in my freezer and I'm about to do these next 2 weeks. But sadly, due to the COVID, I had it all ready to work about a year ago. So that's something that I'm happy to show you, hopefully in the future. So looking at the microbiome is to really look at the microbiome changes following stress within these animals as well. And that would be interesting to see how the microbiome has actually shifted in response to the stress through these different generations. Now, the idea then is that we've got this altered maternal care through the transgenerational transmission of stress. So one thing we could do is if you've got this intergenerational model, we can give them vasopressin or oxytocin. Now, what oxytocin does is this is an important hormone for maternal behavior, and this is reduced in these animals following the chronic social stress. If we increase this level, what happens is that we increase the maternal behavior of the animal, and all those generations downstream don't show that levels of stress and cortisol that we showed you before. So this can completely be reduced by simply targeting the maternal care. I've also been looking at try to understand what other mechanisms are involved in this transgenerational inheritance of this maternal care. And I've been doing some RNA-seq. So we see in the RNA-seq in the F1 animals, we have a number of genes within the hypothalamus and the PVN that are involved in various receptors and calcium binding proteins. And in the F2, we see a number of genes involved in particularly antioxidants. But interestingly, between the F1 and the F2, we have a number of shared genes which are constantly upregulated or downregulated in these animals. So we're having a transgenerational inheritance of some genes that are expressed to the next generation. And particularly ApoE was quite interesting, and a few other genes involved, and the S100, which is involved in a blood-brain barrier, and a few other genes involved, particularly in immune functions as well, and also in neuroreceptors as well. So I think the intergenerational inheritance of stress is very important. If we can understand that, then I think we can tackle a lot of some of the stresses we see. So, for example, there's growing evidence that mother's exposure to early life stress, her exposure to stress, increases her child's risk, her prospective child's risk in the future for behavioural problems. And this is sort of quite recent research coming out to show you the generational effects you can have by early life stress. And that also maternal mental health, particularly postnatal depression, may be a key mechanism by which early adversity is transmitted to children as well. So those mothers with early life stress have increased risks for having children who show stress and depression as well, and even to grandparents as well. We've seen this multi-generational. So I think this is a key area that could be tackled in mental health research. So the whole idea then was that maternal social stress is highly important, and we've shown you that in the animal model. So we've been now looking at in human studies of maternal social stress as well. And a key social stress is something like, as you see here in a study here in the Latina women. So what we did was we looked at the stress of perceived prejudice, and what we did was we looked at this Latina cohort here. And so what happened is that we've these Latina cohort were basically pregnant at the time when Donald Trump came to power, and obviously there was obviously a slight shift in some of the social environment for some of these women. And what we did was that we looked at maternal depression following the social stress, and we found that there were strong links between perceived discrimination in these individuals and their levels of maternal depression. And so looking at the same genes, we can look at the glucocorticoid receptor, and we see that there is increased levels of glucocorticoid receptor methylation in those women reporting perceived increases in perceived discrimination and also acculturation, and that this actually correlated with changes in maternal depression in those individuals as well. We've also seen changes in immune genes as well, and so the immune genes are particularly linked to inflammations, tightly linked to stress, and we see again discrimination scores directly correlate with increased levels of methylation in genes, particularly important in inflammation. And what was interesting with this is that if you divided those women up in regards to epigenetic levels of immune genes important in inflammation, particularly TNF alpha and FOXP3, which is important in T regulatory cells, we could see that those mothers with epigenetic profiles showing reduced inflammation were more resistant to the effects of discrimination as well. And so this is a nice mechanism by which this epigenetic profiles could actually change your risk for developing diseases following environmental stresses. You've also looked at oxytocin, which is so used important in maternal care, and you also see changes in oxytocin receptor following acculturation stress and also everyday discrimination stress as well, which links to oxytocin plasma as well. So this could be the link behind maternal care, linking to the social stress in these mothers. So I'll go on a bit quicker now, so I'm going a bit out of time, aren't I? So some of the studies I'm looking at are in the rural study I showed you in Liverpool, and we've also got a study in Bangalore as well. So what we're looking at again is looking at mothers' mental health and the children's mental health, and we're collecting and looking at the same ideas and testing the same hypotheses I've shown you from the animal models as well. Some of the key genes, and we're also looking at next generation sequencing and going genome-wide on epigenetic profiles as well. Another thing we're looking at is epigenetic clocks as well. So the whole idea about you have the epigenetic clocks of aging, this is something we're working on at the moment. So you have epigenetic clocks of aging that predict more later life diseases. But the idea is that stress is highly important. As I've shown you, stress is important in regulating inflammation as well. And the idea is that some of these epigenetic clocks of aging could be changed by stress as well. And we're looking at that at the moment and developing new epigenetic clocks that could predict environmental stresses and also predict outcomes. And the final 2 minutes, if it's okay, is a major area I'm very keen on at the moment. Is, of course, we know that everyone exposed to stress, not everyone exposed to stress, will develop depression. People can have relatively similar early life stresses, but not all will develop depression later on in life. So the whole idea is, and not everyone with depression has had early life stress. Although it's a risk factor, it's not 100%. So the idea is that there are genes involved. We have different genetics which enable us, which change our responses to the environment. So one of the very famous papers, 20 years ago now, was this gene environment paper. So, for example, if you had a version of the MyoA gene, an allele of the MyoA gene, and you had a low activity, you will have actually low antisocial behavior if you've had no childhood maltreatment. But if you've had childhood maltreatment and you've suffered early life stress, then that low MyoA allele suddenly becomes associated with high levels of antisocial behavior. So it's not just the gene, it's the gene and the environment, and that gene regulates how you will respond to that environment. So what I'm looking at the moment is this recombinant inbred mouse strains. So it's called BXD, and there's a few other inbred strains as well. So what the idea is that these are really looking for QTL. So you have these animals crossed between these other animals here. So there's a couple of different lines, but BXD is the easiest to show you. And that we have a mixture of different lines through breeding for a particular genome. So BXD1 will have some genes from the C57, some genes from the DBA. BXD2 will have different genes that it will share, and they all, across 160 different genomes of pure breeding animals, they all contain different genes in different combinations. So what you can do is you can look for QTL mapping, for example. So if you see these two animals here have high levels of stress, you can see what part of the genome they share. So this part of the genome here in this chromosome, for example, chromosome 4, for example, would suggest there's something in this chromosome 4 which causes high levels of stress in those mice. Now that's been done. But what would be interesting is in stress-related behavior. So what I've done is I've taken some of the phenotype behavior from these animals. And if I look at a tail suspension test and open field behavior, I can see that there is a particular region here on chromosome 2 where there are particular genes that are associating, this region here associates with increased depression and stress on these two behaviors. Within this region, there are a number of different genes, and I've done whole genome-wide methylation to see if there are changes in DNA methylation within this region. And by doing that, I can see, for example, there's one bit of methylation here. And within this region here, we see that there's methylation in that differs between the animals at this region here. And this is the gene here, the SSTR4. And this region here lies directly over an enhancer. And there's a polymorphic difference between these two alleles exactly here. And the SSTR4, for example, is known to regulate stress. So the whole idea is by doing this, I can find out a polymorphism which is important for regulating stress in a whole genome-wide approach, which is very difficult to do currently. So the whole idea then would be then to look for identifying G by E QTL. So what I do is I take those animals again, which is what I'm doing, and rather than looking at the stress per se, I look at stress following early life stress. I look at depression levels following early life stress. So I'm only interested in those that show the changes. And by doing this then, we can find what genes within the genome actually react to early life stress to cause depression. And that's something I'm doing at the very moment. So sorry, I've gone over a little bit more than I wanted to. So thank you for listening. And the whole idea basically is the hypothesis-driven research that I'm very keen to do. So the idea is that animals are crucial for human research. So none of these hypotheses I would get without animal models. And then again, some of the hypotheses I pick up from human research, you need the animals to really understand the mechanism for how these things are working. So the whole idea really is this hypothesis-driven research using a translational approach between animals and humans. So I'd like to thank all the people that I'm collaborating with, and okay, just one plug. If anyone's looking for a genetics book, then I've just published this book earlier this year. So it's really it's about teaching genetics, but using famous examples. So anyway, so I had to get that plug in. Are there any questions? And I will try to.
So thank you for the informative lecture, and seriously, I was able to identify most of the stress in people's behavior because of this as well.
That's
I
good.
mean, it's hereditary in a way, in a sense, if a female parent has been under very much stress, I've seen their child's aging more fastly, more rapidly. I mean, there's hair whitening, hair loss. They aren't even at the age of 25, 24, including myself. I'm having hair loss as well. So yeah, there you have it.
Parenting is a big stress on the parents as well. Yes, absolutely. Yeah, I mean, I would argue possibly that stress probably underlies most chronic diseases we have today.
Yes,
to most, it's incredible the impact that stress has, which people I don't think are particularly understanding. And I think especially with some of the COVID as well, we are seeing variations, and I think stress is having a big role in so much.
Definitely. Are there any questions? Please feel free to ask.
Can I ask a question?
Yes.
Hi, Dr. Christopher. That was a fantastic talk. And I'm more interested about the genetic and the haplotype studies which you were doing. One thing which I wanted to ask is that I have just defended PhD in neurological disorders, that the pups which were subjected to the stress early on in their lives did develop any kind of symptoms of neurodevelopmental disorders or the thing, because they had the stress early on growing. So they might be having some kind of a feature. So if you are having a mouse model, that would be very much elaborative in the lab and evident as well.
Yeah, that's a very interesting question. Was this the transgenerational or was this the mouse one? Because both of them have some different features, and you're correct. Some of the neurodevelopmental features are. I was rather more blinded to focus on HP axis sort of related behaviors. So I was very much focused on depression and anxiety, and I was also very much focused on a few key animal sort of tests really as well. And certainly if I'd broadened my sort of horizons a bit, and then we could have seen some other effects, perhaps effects related to sort of restlessness and perhaps autistic related effects possibly. That's not something I studied. That's something that would be really important to look at, certainly. I think I made it a bit more simpler than it really is, because again I was focusing particularly on the HP axis genes, but I'm sure that there could well be other autism related behaviors as well, and I think some people are looking at that.
Okay. And secondly, sir, I wanted to ask that when you were doing the haplotype studies, Like, so were you considering spatial epigenetic effects in those haplotypes? Because they can be shared as well. Haplotypes
Yeah,
certainly means to the locus of the genome, but yeah, you
yeah,
got my point. Yeah.
absolutely. Now, yes, that's a key question. So the first two grants I put, I wanted to account for cis and trans epigenetic effects, but reviewers just couldn't get their head around it. So I've dropped that. I simply look at cis effects. But you're right, it's the trans effects that could be very important. So, for example, but that's sort of what it does as well, though, because basically what it's trying to do is trying to look for polymorphisms which affect local epigenetic patterns nearby, and these don't necessarily have to be in the neighboring gene. And so, because I said it's linked to SSTR4, and I assumed that, it could well be that that enhancer is regulating genes far, far away in different parts of the chromosome, or possibly even other chromosomes as well. And that is really important, and that'd be something this model would be great to look at. Originally, I was writing a grant for the trans effects, but I couldn't get it past reviewers' heads, and so I've sort of focused on epigenetic changes next to genes, which I assume are the genes more linked.
Thank you. Thank you. Thank you. That's all from my side. That's all. Thank. Nice listening to you.
Well, thank you so much, Chris, for your time.
Pleasure. Thank you.
Appreciate it. We'll get back to you with some potential collaborators who are working in this domain. Our group focuses on the data analysis, EDS, sort of stuff. So look forward to have collaborations with you